So here we are talking of these. So we are talking of Sanger sequencer. Then this is the Illumina, the original version, right? Then you have the iron Torrent machine. Then you have Nanopore, and you can see the size comparison. Nanopore is mostly of size of a large pen drive, right? And it could be held in hand. And that is the plus or advantage with Nanopore. We'll talk of it in more details as we go along. Then you have the ProMethan. This is the multiple assembly of nanopores. And then this is your Pac Bio sequencer. So this is the evolution of sequencing techniques we're talking about. In between, there have been many other techniques that have come and gone. For example, pyrosequencing is obsolete now. Likewise, solid sequencer is obsolete now. So there has been evolution in the last 20 years. We have moved from first generation to second generation to the third generation of sequencing. But sequencing began with Fred Sanger, right? And Fred Sanger got Nobel Prize twice. One was for identifying the insulin sequence, and the second was for developing a technique for the DNA sequence. So Sanger sequencing, as you popularly know. And the other person here is Walter Gilbert, who also developed a method of sequencing the DNA, which was not so successful because it could not be automated. So this is here. So even today, most of the sequencing techniques rely on polymerase. So what happens is you have a template that you want to sequence here. You add an adapter to it so that you can add a complementary primer here. And once you add the complementary primer, you give DNA polymerase, and DNA polymerase, as it extends the nucleotides, you are able to, in some manner, identify which nucleotide got added. And that allows you to basically decipher the sequence on the template because of the base complementarity rule, right? So here you are, this is how you do it. So basically, most of the techniques rely on polymerase-based sequencing. You have a primer of DNA of length n. So this is the red fragment is represented as DNA of length n. You give a ddNTP and you give DNA polymerase. The primer gets extended by one nucleotide at a time, and it also releases a pyrophosphate and an H+ ion. And these are important because in some cases, you identify by which nucleotide got added. In other cases, you identify by whether the pyrophosphate got released or not, especially in pyrosequencing, this was the case. And in other cases like iron Torrent, you identify the signal of incorporation based on whether the pH of the medium changed or not, because there will be hydrogen ion release if the incorporation of the nucleotide takes place. So therefore, each of these has its own importance, but most commonly what you use is a fluorescent ddNTP, which is differentially colored, which means A, T, G, and C are all differently colored, and that allows for basically, identification of which nucleotide got incorporated in real time. So we come to first the Sanger sequencing. Very quickly, we'll go through this. So Sanger sequencing was a very smart way of sequencing the DNA. They use ddNTPs, and alongside they also use what is known as ddNTP or dideoxynucleotide triphosphate. So these would not have a 3' OH, and because the 3' OH is absent, then once the ddNTP gets incorporated, further extension does not happen. So here you are, you have your primer and you're extending. So the polymerase will have a choice of adding a normal thymine or a dideoxythymine. If a dideoxythymine gets added, no further extension happens. However, in cases where you have a normal thymine, it moves to the second position. Again, it has an option of adding a dideoxyadenine or a normal adenine. If a dideoxyadenine gets added, you will not get further extension. So likewise, you keep doing on these positions, and what you end up is, if you see here, what you end up with is a series of fragments that differ from each other by one nucleotide, and they have a color tag at the last nucleotide that is there. And because each of these 4 nucleotides have a different color, you are able to identify what is the last nucleotide in this particular fragment. So what you do next is you separate them out on a gel, and the gel that we use here is capillary electrophoresis. And then, of course, you also observe what is the nucleotide passing through at the point of observation. Based on the color, you have a laser that detects what is the color here, and that allows you to translate it into a language of nucleotides, and that is how you get a final sequence. So this is your sequence here. If you see, what you get is a chromatogram, and which corresponds to bands, and these can then be, translated into actual nucleotides. So automated Sanger sequencing was a smart move because it used differentially labeled fluorescent ddNTPs, facilitates resolution of all fragments in a single lane instead of 4 for each sample. So before using differentially labeled ddNTPs, the only label available was what is known as the radio label, and radio label would produce black color. So therefore, for one sample, you had to do 4 different reactions. With ddNTP, differentially labeled ddNTPs, it became easy. For one sample, you had only one lane, and that became easy. Differential labeling also increases the read length, as the bands can be allowed to flow out of the gel post-detection. So in this case, you could not allow the gel to the bands to flow out of the gel. Here in this case, you have a detection point here. Once the band has been detected and the color is known, you know which nucleotide it corresponds to. You can allow it to pass through and allow the other bands to get resolved, right? Then you use capillary electrophoresis and higher parallelism of 96 samples in one run, and the sequence output is in the form of a chromatogram. So this is typically a chromatogram that you see here. And you had automated Sanger sequencer, and automated Sanger sequencers were, while they were, very effective in sequencing the human genome. So the major achievements of these Sanger sequencing, in 1995, we were able to sequence the first free living organism, that is Haemophilus influenzae. In 1996, we sequenced yeast. In 2000, we sequenced Drosophila and Arabidopsis. These are both model organisms, I'm sure you know. Drosophila is for animals, this is for plants. And then in 2001, the crowning glory of Sanger sequencing was that we could sequence the human genome. However, the problem was that it had taken us 10 years to sequence the human genome, also because there was no reference available earlier. And then, of course, what was more important was that besides 10 years, it had taken us $3.2 billion to sequence the first human genome, which was a problem. If a genome was to be sequenced at $3.2 billion, we are not anywhere close to our idea of personalized medicine, right? So therefore, there was a grand challenge that was launched, and that resulted in the second generation of sequencing techniques, and developed to sequence and assemble the whole genome using a reference. Speed and cost effective was what was important. The idea was to develop a sequencing technique that would sequence a human genome around $1,000, right? And very recently, we have come very close to sequencing the genome in $1,000. So based on these techniques and the evolution of sequencing techniques, you now have 3 generations of sequencers. One is the classical, the first generation. We've talked of Sanger method. Then you had pyrosequencing, virtual terminator sequencing, and solid. Both pyrosequencing and solid are now obsolete, but this one here by Illumina, virtual terminator sequencing, is the market leader even today, right? And then, of course, you move on to the third generation. This is 2008 and evolving. And you have Nanopore and Torrent and single molecule real-time sequencing, which are now leading the market. Illumina, of course, is the one that leads the market today. 80% of the market is by Illumina, but the long-range sequencers are fast catching up with it. So here you are. If you look at the comparisons, the effort has been into increasing the read length. And if you see here again, the pyrosequencing was a polymerase-based sequencing. Solexa was polymerase-based sequencing as well. This is Illumina. And solid was ligase-based sequencing. Now these are obsolete. Illumina is what continues. So if you look at the contemporary sequencing technologies that are available in the market today, you can classify them into 2 different categories. One is what is known as the short read sequencers, and the other is what is known as the long read sequencers. And as the name indicates, short read sequencers will have short read length, and therefore the assembly would be a bit of a problem. Long read sequencers will have longer read length. The assembly will not be a problem, but there could be problems in the actual accuracy. So what people now do is to go for long read sequencers to ensure that they get the correct assembly, and basically couple it with short read sequencers so that the individual positions, if they're wanting to look at variations, those are basically correctly identified by short read. So you have a hybrid, methodology, or basically synergize long read with short read to get correct assembly and also the precision in terms of the individual variation points that you're looking at. You follow class, everyone?
Yes, sir.
Yes, sir.
Okay, good. So then we move on and we talk a bit about common features of second generation sequencing. So here you are, library preparation, random fragmentation starting DNA, addition of custom linkers. This is common to all of the second generation, whether it is pyrosequencing or Illumina or solid. Then you have the library amplification. So the individual fragments that we generate, they have to be amplified in a local, very closed area that is basically called a cluster. And there are 2 types of PCR that we do here. One is called the bridge PCR and the other is called the emulsion PCR. Bridge PCR works for Illumina, and the emulsion PCR works for pyrosequencing, for solid, and for ion torrent, right? And then Next, what is important is that all of these techniques give you a direct detection as the nucleotide gets incorporated. You have an idea of which nucleotide got incorporated. So this is real-time sequencing. And the other important feature is, and where it scores over Sanger sequencing, is what is known as the massively parallel sequencing. So these are high throughput techniques. You have millions to billions of reads coming out from one run of the machine. So therefore, the short read is compensated by the actual output that is very huge. In contrast to capillary sequencer, that is the Sanger sequencing, the read lengths are shorter here, right? So I have already told you, Illumina will give you 150 bases, and your ion torrent will give you around 100 bases, right? And pyrosequencing could give you around 400 bases, but now it is decommissioned, not in use anymore. So first we talk of Illumina, or the basic sequencing reaction that takes place in Illumina. And Illumina has a whole range of, so this is basically your revolution where you have, depending on what is your requirement, you have various types of Illumina sequencers giving you a certain range of high throughput depending on what is your individual requirement, right? So this is also democratized the sequencing revolution. Earlier, sequencing used to be only restricted to a few centers, and you have to send your samples. Now, individual labs can have sequencers of their choice based on the requirements. So this is basically also democratization of the sequencing reactions that were initially not possible. So that's the revolution. So here you are. What Illumina uses is a technique very similar to Sanger sequencing, except that in Sanger, the termination was permanent. In Illumina, the termination is only temporary. So what they use is proprietary nucleotides that have a virtual terminator moiety and a fluorescent molecule attached to it. As long as the virtual terminator moiety is attached, no further extension happens. That gives you time to identify which nucleotide got incorporated. Once you have identified the signal, you cut off this virtual terminator moiety, and now it is ready for the incorporation of the next nucleotide. And I'll show you an animation how this happens in the next slide. So the virtual terminator nucleotides are nucleotide analogs, contain a fluorescent dye and a chemically cleavable group called virtual terminator group or virtual terminator moiety. Once incorporated, the virtual terminator analogs block further incorporation until the virtual terminator moiety is chemically removed. Therefore, they're called virtual or reversible terminators because the termination that happens is reversible in contrast to Sanger. The virtual terminator label is removed before the next cycle, and you could do it by chemical treatment. So here you are. So here is your flow cell. I'm showing you only two templates. Actually, these are multiple copies of the same template, millions of copies of the same template in this location. Likewise, another template here, and millions of copies of this here, which are generated by bridge PCR. And now you start your sequencing reaction. You give all four nucleotides together based on the base complementarity rule. Individual nucleotides will go and anneal. And because they are differentially labeled, they would give you a color. And this color is what you're detecting as your signal, right? So here you have a thymine incorporation. Thymine is blue, so you know on the template there will be an adenine. Likewise, here there is an adenine incorporation. Adenine you have tagged as red, so now you know that on the template there is a thymine. And this is your virtual terminator moiety. So before moving on to the next cycle, you need to remove your virtual terminator moiety. Once you remove that, now you move on to the next cycle. Again, you have the incorporation based on base complementarity rule. And then again, you have the terminator moiety. So therefore, no further extension happens. You can read what is the fluorescence coming at this position in the flow cell, what is the fluorescence coming at this position in the flow cell. That allows you to identify the nucleotide incorporated. And then again, you can remove this and move on to the next cycle. So this is how it happens. This is how it works. So we are looking at two positions here, position 1 and position 2, and we're looking at some 6 cycles here, right? So if you see here, when you look at this the first time, you get the green signals for both, which means the cytosine got incorporated. Then the second time, you have adenine incorporated in one, and you have a cytosine incorporated again in the second one. And you can keep doing your cycles, and then you keep observing the colors that are obtained at individual positions. And that finally gives you the sequence. For example, here, the second sequence, every time it is a green that is getting incorporated, which means every time cytosine gets incorporated, which means on the template, it would be a guanine, right? And for the top one, you have for every cycle a different color getting showing up, which means a different nucleotide gets incorporated. So finally, you have a sequence of CAT, CGT, right? So this is how you take your cycles. And if you see the actual file of FASTQ file of Illumina sequence, you will see this term cycles. Cycles means how many nucleotides have been incorporated, right? So as many cycles you grow, as many cycles will be nucleotides would be incorporated. Next, we come to the next generation sequencing techniques, the third generation. Here there is no imaging, and because there is no imaging, these are faster and much cheaper as compared to the second generation. And here we have one that is ion torrent, and this is an ion torrent machine here. And if you look at the basic idea of ion torrent, I showed you in the incorporation of a nucleotide via DNA polymerase. As the nucleotide gets incorporated, you have the release of a hydrogen ion. So instead of checking for fluorescence, this machine checks for whether there has been a decrease in the pH upon incorporation of one type of nucleotide, right? So what you do here is, instead of giving all 4 nucleotides together, you give only one type of nucleotide in one cycle and look at positions where there is a decrease in pH. So that is the whole idea behind ion torrent. So here you are, the flow cell is flushed with single nucleotides at a time. The semiconductor chip records pH changes upon incorporation of complementary nucleotide by polymerase. So this is important, that you give only one type of nucleotide at a time. You know what you're giving. So therefore, if you're giving adenine and you see at certain positions there was a decrease in pH, you know that there was a thymine on the template there, right? The change in pH is proportional to the number of nucleotides added, and current read length is around 100 bases. There has been some improvement to this, but not beyond, let's say, 150 bases for now. Now we come to the new range of sequencers that have come up, and these are the long range sequencers, and one of the primary ones here is what is known as Nanopore. And the advantage of Nanopore is it is small in size, so it can be used as a point of care machine, a point of care device for a country like India, where the hospital setup is not very expansive, and patient has to go to the hospital and get checked. These devices can help the individuals go to the patient instead of patient coming to the doctor. So basically, these can serve as what is known as the point of care device, right? And that is the advantage with Nanopore devices here. So what is a nanopore? A nanopore is essentially a nanoscale hole. The hole may be biological, solid state, or hybrid. And the idea, and here is an example of a biological nanopore. This is alpha hemolysin protein. It is a haptomeric protein, and you can see the 3D structure here. This is the tunnel. This is actually the nanopore, and this is the side view. This is the front view. This is the tunnel through which you can pass your DNA if you wish to. So the idea behind nanopore sequencing is very simple. The nanopore is placed into an electric field, and because it is in an electric field, a basal current passes through the nanopore. Now, when you introduce obstructions to the nanopore, or you allow certain molecules to pass through the nanopore, based on the shape, size, and charge of the molecule, you will have characteristic fluctuations in current. And for us, luckily, what you have is, depending on both adenine, cytosine, guanine, and thymine produce different fluctuations in current. So that fluctuation in current is characteristic of the nucleotide that is passing through. That allows you to translate it into a sequence, right? And what is also important here is that even a methylated cytosine will produce a different fluctuation as compared to a non-methylated cytosine. So therefore, here you can also study the epigenetic state of your genome without going through a very complicated process of bisulfite sequencing, which is done in Illumina process, right? So this is where the advantage is. One, you get long reads. Second, you can also get a readout of your epigenetic state of the genome without any special treatment of the DNA, right? So when a single molecule passes through the nanopore, characteristic disruptions occur in the current flow based on the structural features and charge of the molecule. So this is what you do. You basically use a helicase. So this is the technique where we are not using DNA polymerase. So remember this. So in nanopore, you're not synthesizing a complementary strand and reading the nucleotides as they get incorporated. What you simply do is unwind a piece of DNA and force it through a nanopore, which is placed in an electric field. Based on the fluctuations in current as the nucleotide passes through the sensitive position, you will have the sequence translated into ATGC, right? So this is what is Nanopore, Oxford Nanopore, marketed by Oxford Nanopore. Time to sequence human genome would be around, roughly 1 hour or something, 15 to 20 minutes if it is absolutely perfect. A size of the sequencer is a pen drive, and this is scalable. You could have multiple nanopores put together into an assembly. Cost of sequencing the human genome would be around $1,000 here, right? So nanopore advantages: direct RNA DNA sequencing, real-time, ultra-long reads. I've already told you these are long read sequencers, and you could get ultra-long means around 50 kb of read length. Direct methylation call, you don't have to go through a bisulfite process for sequencing. scalable to portable to or desktop. And then, of course, point of care device. You could have you have a very small average preparation of 10 minutes and the high fidelity. This is basically the advantages that you have. The other one that we have is what is known as SMRT, single molecule real-time sequencing. This is still relying on DNA polymerase. But what is important here is that it's only taking a single molecule for sequencing. So what are PCR artifacts that may happen during your Illumina processing or for that matter, ion torrent, which also uses PCR? These are ruled away, right? These are outright ruled. Now, there are two challenges to a single molecule real-time sequencing. One is the signal intensity. So because you have only one molecule and you're incorporating a fluorescent ddNTP, the signal intensity will be very low. And how do you compensate for that? Because that could also be an artifact or a complication. So for that, what you do is very a simple idea. Let's say you have a 40-watt bulb and you are placing it in your drawing room, which is the largest, into your kitchen and into your bathroom, which is the smallest. So you know the signal will be, the intensity of light will be the largest in your bathroom because the area that it is covering is very small. So likewise, what SMRT does is to basically immobilize a piece of DNA polymerase and your DNA in a very small volume, which is known as zero wave guide, right? And we'll talk of it as we go along. So what you have is zero mode wave guides. So these are very small spaces in which a single DNA polymerase is incorporated, and it is then basically it's provided with the DNA template that you want to sequence. So therefore, the intensity of signal is still good enough to detect because the volume of the space where you're doing the reaction is very small. Then the second important problem is polymerase fidelity. So you know that polymerase is not perfect. So while you're sequencing, it may add a non-complementary nucleotide as well. So if you have a single molecule, then once misincorporation happens, you would have this idea that, you know, this cannot be corrected. So what they do here is they circularize the template that you want to sequence. So this is the sequence template that you want to sequence. You circularize it by adding adapters at both ends, and then you sequence it multiple times like a rolling circle model. So the polymerase keeps sequencing it multiple times. So what happens is you will have the entire sequence, and in between you'll have your adapters. Then you align your sequence that you get. So this is kind of a concatemer that you get, and you align it together. And for each position, you look at the most frequent nucleotide that is there. And that is what is known as a hi-fi read. You generate a consensus sequence depending on what is the nucleotide most commonly occurring. That is your greater than 99% accuracy now. So you have overcome the two basic problems that are there with single molecule sequencing. One was the signal intensity by creating zero mode waveguides. And second, you are looking at, you know, circularizing your DNA and sequencing it multiple times. So if at all there is a problem with respect to the fidelity of polymerase, you're still looking at a consensus sequence. So the nucleotide that appears most commonly at a given position is what is your final sequence. So this kind of gives you high accuracy. So these are known as the hi-fi reads. So this is your system here. And then of course, this is a comparison of read length. So you get on an average half the data in reads, so greater than 30 KB. Some of the reads, top 5% reads are greater than 50 KB and so on and so forth. So therefore, you know, these are long read length, and that allows you to basically assemble the genome more correctly, and a de novo assembly of genomes is possible.